given our mission to maximize health span, I couldn't be more excited to introduce our special guest today, Professor Nir Barzilai. And he's asked me to call him Nir as opposed to Far, so I will call him Nir. Nir is the founding director of the Institute for Aging Research, or AFAR. He's the director of the NIH Nathan Schock Center of Excellence in the Basic Biology of Aging. And he's the director of the Paul F. Glenn Center for the Biology of Human Aging Research at Albert Einstein College of Medicine. He is also the chair, the Renner Chair of Aging Research. He's a professor in the Department of Medicine and Genetics, and he's a member of the Diabetes Research Center. And I think to this day is still seeing patients with diabetes in that center, as well as a professor in the divisions of endocrinology and diabetes, as well as geriatrics. Nir also holds an NIH Merit Award, which focuses on the metabolic decline associated with aging. And he's currently leading an international effort to approve drugs that can target aging. And this is pretty exciting. And so one of these efforts is known as the TAME study, which I'm sure he's going to talk to us about today. It's an acronym for Targeting Aging with Metformin. So this is the first and the largest study of its kind. And I believe it has the potential to be one of the most important studies in medicine. Metformin is a drug that is 100 years old, actually, this year. I think it was discovered in 1922. It was introduced as a medicine in France in the 50s for influenza, for malaria. And it was then found to actually lower blood glucose. And that's when it was introduced and approved by the FDA in 1995 as an anti-diabetic medication. It's now the fourth most commonly prescribed drug in the United States. And I believe, and Nir will correct me if I'm wrong, but the cheapest drug on the US formulary. It's also on the World Health Organization's list of essential medicines. It's the most commonly prescribed and used oral anti-diabetic medication in the world. And now there are billions of patient years of use with this drug. So we know a lot about this drug, but we're going to learn a lot more about it. So Nir, I can't thank you enough for taking time to be with us today. We're really looking forward to your presentation and looking forward to collaborating with you in the future. Take it away.
Thanks, and I have nothing to add. You summarize it very nicely, and we'll open it to questions, right? So it's really good to be with you because the little I know about Atria, and I read, and then you gave me presentation, I'm trying to build the future, but you're already in the future. And I think we have to do things in parallel. But I think also we have to a little bit coordinate. So it's really a pleasure to be here. And before I share my slides, a few things I just want to say. First of all, before you leave this presentation, if it's going to be very boring for you, I just want you to know that there's one thing that I want you to take away, and that is that there's a biology of aging. No, you know that, you know who's old and who's young, but really that this biology of aging drives diseases of aging, okay? You can be born with an ApoE4 genotype that will make you most probably demented at some point, but you don't get it when you're 1 year or 10 year or 20 year or 30 years old. You need the biology of aging in order to get this disease. But the most important thing about the biology of aging is that it's very easily modulated. And in my labs and in labs around the world, we modulate aging quite easily. And we extend health span, which is the most important thing, and lifespan. It's much easier than to treat cancer, so I think there's a promise in preventing medicine in aging, and I'm going to exemplify just some of the science from my point of view. The second thing that I just want to throw out, I didn't mean it until I talked with you guys before, and that is that by the way, AFAR is the American Federation of Aging Research. It's not the Institute for Aging Research at Einstein, but I'm the scientific director of AFAR. I'm also the founding and the executive of the Academy of Health and Lifespan Research. And lastly, before I share my slide, I'm a geroscientist, okay? So I'm not in anti-aging, okay? That's a noise for most of us. I'm a geroscientist, and geroscience is there in order to discover geroprotections and gerotherapeutics Okay, so I'm going to talk a little bit about the aspects and the challenges in that. And now I'll really hope that I have a more exciting part of my presentation. This was so much more administrative. So, yes, you know, aging has really bad connotation, and I think that few people understand that yeah, death is inevitable, but not aging the way it is. And really, over our 50,000 or 100,000 years of human evolution, our life expectancy was somewhere between 19 and 35. There were always some old people, but it's only in the last 150 years, so it looks such a steep increase in the time scale, that we achieved this average age of 78. And I want to just make sure that you know that some of the things that allowed it are preventive, okay? By how we cultivated food, how did we deal with water for agriculture and water for drinking, sewers, vaccination, and some interventions like antibiotics and surgery, okay? But it was a lot of prevention. And then we got to an average of 78, and we realized that we did some bad deal here because all of a sudden we got an age-related diseases like Alzheimer and cancer and diabetes and cardiovascular disease, and they're accumulating at the end of our life, and they're giving bad quality of life. And I think now that everybody realizes that it's been there for those whole last 100 years, there's something that we need to create now, a new history, and we can create a new history about it. And it's really about prevention and the question of whether this aging can be prevented, and because of that, can we prevent those age-related disease, their accumulation, and the bad quality of life. As I said, I'm a geroscientist, and I think what we've done with geroscience was kind of interesting. We all agreed that they are hallmarks of aging. We have internal fights of what to include and what to exclude, and people are having their own slides, but pretty much we agree that they're hallmarks of aging. Now, how do you become a hallmark of aging? You have to show that it goes bad when you age, and that if you treat it or prevent it by genetic tools, by drugs, but however you want to do it, that you get an improvement. The interesting thing is that those hallmarks are interrelated between them, and that's very important because you don't have to treat all of them in order to have effect. You can do one of them and have effects on others. And that's really quite incredible. And it's really important because now that the biotechs are in, what they're trying to use is to say which hallmark I want to target, knowing that it probably is going to have an impact on other. And what we've done in the field is, although we don't know everything that we need to know about aging, we did discover some pathways that lead to longevity. And because of that, we have really a good head start. And we increased health span and lifespan in numerous animal models, mainly in mammalians, which is really the most important. And there are some gerotherapeutics that are available now to humans. This is a challenge to repurpose that, and I will touch about that soon. So those are the hallmarks of aging, and their interaction is very important, and I'll touch about some of it later on. So what I'd like to do from here on, and by the way, I have 40 slides. It usually takes me 40 minutes, but you relaxed me, so it'll take longer, but I'm really eager to go and answer your questions. So hopefully in less than an hour, I'll be done. So I want to talk about human longevity genes with the same passion as people are talking about cardiovascular genes and cancer genes and other genes that are associated with diseases. The second topic I want to cover is biomarkers. We had a great lecture by Steve Horvath the other week. And I want to make some other points because we heard more of methylation of clocks and the challenges there. So I want to kind of give another layers to that. And the third part is to talk about the regulatory challenges of repurposing drugs or repurposing gerotherapeutics, and really talk about TAME and Metformin. So those guys here in the picture were born in New York between 1910 and 1920. And what's incredible with them, all of them reached beyond the age of 102. So she was 102, 107, 109, and 110. Okay, what are the chances that four siblings born to two people are all becoming centenarians? We have almost 750 families of centenarians, and we have also a longitudinal study of their offspring, okay, to see the effects of the longevity genes on the aging of their offspring. So there's a lot of things that are going on in this study, but I'll just pick some points that I think will be interesting to many of you. And really the first thing is, the question was, are they getting sick when everybody gets sick? And what they do is they live sick longer, or is their health span and lifespan, does it go together? And you can see in this slide, we have two groups of centenarians. The New England centenarian studies, those are black and red, and our study, the Longevity Gene Project, which is green and blue. The green are controls, the blue are the centenarians. And what we are measuring here is disease-free survival. So everybody is healthy initially, and basically after the age of 60, I'm doing the green and blue line because that's my study. After the age of 60, they're basically accumulating those diseases. And after the age of 80, just like 10% don't have any disease. Centenarians live 20, 30 years healthier. And even at age 100, almost 30% of them don't have a disease. A lot of them just are not waking up in the morning. And actually, this is really the important part. It's not only that lifespan and health span goes together in those people. Okay, so we have example of our potential, but it's not only that. It's they have a contraction of morbidity. In other words, they're sick very little time at the end of their lives. And this is very important for us, again, in the politics of things, because the CDC, and nice, now everybody knows who's the CDC, have monitored the cost, the medical cost in the last 2 years of life of people who were born, of people who died when they were over 100 or when they were in their 60s, 70s, and it's third the cost. This is when they started, it was, I think, 1993, and it's still the same now, which led to this whole discussion that's called longevity dividend. You know, how many trillions of dollars can we save if we can just imitate, not even centenarians, but increase the health span but by a year or two, very modestly. And recently, Andrew Scott, who's a professor of economics in London School of Economics, have said, guys, you are so way off. You have no idea what you're talking about, because this is medical expense. But you forget, if those guys are not in the hospital, what are they doing? They're traveling, they're shopping. They add value to the economy much beyond not being sick. And he came and recalculated, and the calculation is of like $500 trillion in just several years. I think we cannot afford not to target aging, delay and improve health span. I think that's the economical point that we have to use and use more often. So back to my academic interest. So you could hypothesize that there are three possibilities of why those centenarians are alive for so long. One is they interacted with the environment. They knew what we know now all their lives. So they exercised, they checked their diet, they did all the right things, and that's why they got to be 100. And I'm not going to show you data, we published that all, but actually 60% of the men and 30% of the women were heavy smoker. Remember in my picture, Helen, who got to be 110, she stands there on the left side. When I met her, she was 100 years old, and she was outside her door in a New York apartment, and she was smoking. And I said, Helen, nobody told you to stop smoking? And she said, all 4 doctors that told me to stop smoking, they died. Okay, so it's in spite of the environment. 50% of them were overweight or obese. 50% of them didn't even move. I mean, mild things like housework, walking, biking. Vegetarians, 2%. Okay, so the point is, there's nothing good about the environment. They in fact showing us how those genes have protected them against bad environment. If you ask a typical centenarian what was his diet, he would immediately tell you the one thing that he shouldn't have done, okay, like eating chicken fat every day or something like that. Okay, so there are 2 other possibilities. One is that they have the perfect genome. I mean, we know all those SNPs, all those genetic alterations that leads to cardiovascular disease, Alzheimer, cancer, diabetes. So maybe it happened that 1 out of 10,000 just don't have any of that. So, or the third alternative is they have longevity genes that are actually slowing their aging. So let me make just one point about the perfect genome hypothesis. And this is one of our first study because this is whole genome sequencing in our first 44 centenarians. Okay, so there's no control here. Okay, only 44 centenarians. And we pretty much asked how many other bad genotypes they have. And there is this database that at that time had 50,000 variants that are pathogenic variants, which meant they most probably will give you a disease. And we said, if they have perfect genome, they should have very few of those variants. We certainly were not ready to see that 44 centenarians have almost over 230 variants that should have given them disease, and they're 100 years old and don't have this disease. Okay, so they can harbor not only a bad environment, but a lot of other junk. And if you're asking, are they important genotypes? They're very important genotypes, okay? Like the ones that are pretty much predictive on Parkinson. We have 2 centenarians who have those ApoE4 genotypes that I told you they would be, demented at 70 and dead at 80, and they're 100 years old and not demented. Degenerative disease, neoplastic disease, cardiac disease, dominant disease, recessive disease, and other. So no, so it's not that they have perfect genome. They probably have mechanisms that slows their aging. So Zhengdong Zhang, who is a geneticist but with a computational approach, has taken our data of basically whole genome sequencing in all our 3,000 people and really did something that I think overcomes a mistake that we've been doing. You know, in genetics, we look at one SNP at a time. We ask, is this SNP out of thousands or millions or whatever, is this SNP associated with the disease? But we're not built from one SNP. We're built from many SNPs, and many SNPs will cancel each other. So what he's done, he started by looking at very rare occurrence, less than 1%, okay? And looking at the difference between centenarians and people without longevity, okay? Which we were all doing. But it didn't stop there. he went and put those SNPs into pathways, okay? Because as I said, there are pathways with lots of proteins, and you want to see if things are enriched in pathways rather than enrich in a single genotype. And he also used some prior knowledge in some of it, not in the slides that I'm showing you now. So basically, if you look at the pathways, what you're getting as most significant pathways is exactly the things that we knew already. Okay, mTOR, right, where rapamycin acts, or insulin signaling pathway, IGF signaling pathway, which is where IGF and insulin acts, and MET kinase signaling pathways, and there are several other pathways that are involved like that. And I think for us, it was important, first of all, to validate, unlike other animal models, aging animals are giving us very good model for aging. And actually, the problem, if any, is the fact that a lot of people who are doing studies on age-related diseases study them on 2-month-old mice. That is totally not the background of the biology of aging. And we have this fight, actually a successful fight, because a lot of the institutes now would give you money if you want to do your studies on aging animals. But what we knew from aging animals and what we know about longevity and what we know from humans is pretty much the same. So that's good. But I want to give you a specific example, okay? And the specific example is about growth hormone and IGF signaling pathway. And the reason is that you know that small dogs live longer, the ponies live longer than the thoroughbreds. When you mutate this pathway in lower organism, they live longer. If you do transgenic with lots of growth hormone, or you knock out the IGF or the growth hormone receptor, the more growth hormone you have, the less longevity you have. And in our centenarians, okay, so our centenarians are already 100 years old, and they're likely, their death is 30% a year, pretty much. Okay, so they're at the end of their lives. But if you look at their mortality based on the level of IGF, okay, which is a good marker of growth hormone, peripheral marker of growth hormone, then those with the lowest IGF have twice the longevity of the ones who have the highest level of IGF. In fact, 60% of our centenarians have some genetic alteration in this growth hormone IGF signaling pathway. But by the way, David, if I'm saying something that obviously people will not understand, did I just say a word that people don't know what it is? You please stop me. I would just say, if people not know what IGF, growth hormone, is this hormone that is secreted from the pituitary and increase other growth hormones, and the major of which is IGF-1. So if I miss something, for the for the intelligent but non-specific, David, make a point. We have time for that. And of course, we always want to make sure that that it's the brain and not only the rest of the body. And in fact, when you had the lowest IGF-1 level, you're less than half as likely to have any cognitive impairment. So it's not only related to the level and mortality, it's related to phenotype, and we've done much more phenotype than that. But I want to tell you an interesting story, because if you're tall and you say, "Oh God, I'm going to die soon," I want to tell you really an interesting story. So my then fellow Gilad Smon is an independent professor now. 15 years ago, basically came to me and said, we were looking at all the alteration in growth hormone IGF signaling pathway. And he said there's a common deletion of exon 3 in the growth hormone receptor, a deletion in the growth hormone receptor. And he says, when I'm looking at this expression in population, I see that in most of the people in most of population, it's 4%, but it's almost 12% in centenarians. And there's kind of a dose response for that. So I said, well, great. So what's their IGF-1 level? We're measuring IGF. And he said, well, their IGF-1 level is significantly lower. And I said, that's terrific. And what is their height? And he said, well, that's a problem. They're significantly taller. And for 10 years, I said, you know, I believe all the data, but how can I put a story like that really makes no sense? And he said, what should I do? I said, well, there are two things. The first thing you should do is go and look at other populations and just validate. In genetics, we need to validate everything. And he went and validated in other population, and it's all the same. The oldest old have much more of the growth hormone deletion in the growth hormone receptor. So now it is, well, how do I understand that they're taller? So we collaborated with Hassi Cohen, who I have collaboration since medical school, and he's a growth hormone expert. And we have the cells of our subjects, and we send him the cells of our subjects, and he incubated them without or with growth hormone. It looked at the activation of the receptor. And we found there's something very interesting with when you don't put growth hormone, the activation is half, is very low, consistent with low IGF-1 level. But when we incubated with growth hormone, there was almost a switch that turned on and doubled the activation. And it's not only the activation, it's the proliferation. It's the same, it's lower, and then you give growth hormone and it's higher. So to cut a long story short, what we think happens is they are born with this mutation. And when they go through puberty, which is the only time that growth hormone increased so much, they get activation of the growth hormone and they grow taller. And then after growth hormone comes back, it kind of shuts off and it gives them the advantage of longevity. So biology is always interesting and more complex. And the point here is you still can have tall people, but have low growth hormone because of some biological phenomenon of turning something on and off. Can we do something about this growth hormone IGF? Well, with Derek Hoffman, we coordinated a study where we took IGF receptor antibody that was developed for cancer and failed, by the way. Cancer, I told you, cancer is much harder. Cancer always find a way not to do things. But the antibodies were developed, they were in human trials, and we got from Amgen neuronized antibodies, and we gave 18 to 22 months old mice the antibodies. That's an injection once a week. And they lived significantly longer, 10% longer. But again, it's not about lifespan, it's about health span. And in this paper, we showed that their cardiac function improved, exercise capacity improved, sensory motor function improved. And one of the things is that it's never really too late to target aging. A lot of the trials we start at young age, and I think it's a problem. But when we start some of the treatment at all age, we have even a better effect. So on theory, IGF receptor antibody can be repurposed in certain cases to target aging. So now I'm switching from kind of the genetic of exceptional longevity to the biomarker part. And we talked about methylation. And of course, I think Steve explained very well why we need a marker, not for your chronological age, but for biological age. And for me, it's more important than that. We need a biomarker that will change with treatment and will change relatively fast so that we can know if a gerotherapeutics that we're developing is going to be consistent and likely to change health span and lifespan or its combinations. And as Steve said, with methylation, he said he doesn't really believe it. It happens with less than 9 months or a year or two, he said, that methylation is really much more stable. And what we did is we took our subjects, not all our subjects, but those between 65 and 95,000 subjects from our longitudinal study. And by a new technology, SOMA-MER, that is an interesting technology, we looked at the expression of 5,000 proteins in the plasma, okay? I'm not going into this technology. And now I'm showing you a slide, and let me just explain what you're seeing. It's known as a volcano plot. All of us were dealing with one protein at a time, know how to read volcano plots. Volcano plots are you throw all the data, and the computer spits from the volcano those red lava rocks, right? And the higher they go, it's really it's the statistical value. So for example, those guys here that are up here, their p-value is 10 to the minus 80. Okay, you need 80 zeros because it's so significant. And what you see here from the zero, those are the ones that are increasing with aging, and those are the ones that are decreasing with aging. You see my cursor, David, right? You see my, okay. So that's a volcano plot, and this volcano plot shows you that there are almost 800 protein out of those almost 5,000 that are changing with aging. Some of them are extremely, extremely significant. And now it's a problem, because what does it mean? Those proteins might cause aging, or, you know, when we start this breakdown of aging, they might be protective against aging, right? When we have infection, we have an inflammatory response. There is inflammatory response in aging too. How do we decide, not what's biomarker here, but what do we learn biologically? For example, this protein PTN and MIC, that is also GDF15, when you overexpress them in animals, they live healthier and longer. Okay, so if you thought those proteins we should decrease, okay, it's probably wrong. If anything, I would increase them even more. Okay, maybe, I don't know, but this would be my hypothesis. So this is kind of a mess on its own, but it's not a mess from a biomarker perspective. Now, if you're asking, what are the groups, right? I said that we are looking things and pathways. What are the groups? Then we're doing this analysis, and I'm not going to tell you much about it, although the first ones that are coming out is regulation of IGF. Okay, whatever we do, IGF, growth hormone, they always come through. But there are other things that are really important. And first I thought, this is junk. I just don't want to see it anymore. But it's about extracellular matrix, about degradation of it, the granulation of platelet, degradation of collagen, and neutrophil granulation, collagen biosynthesis. And you know what it reflects? It reflects the breakdown with aging. And I think that maybe no matter what gerotherapeutic you use, no matter where on the hallmarks it's going to hit, what you want eventually is to stop this breakdown, okay? So it's possible that those kind of proteins are going to be the most important biomarkers for gerotherapeutics. There are other things that are interesting, and I just want to show you. And that has to do with the fact that we totally missed sex differences in our studies of biology of aging. In fact, in all diseases, because we had some excuses that we need to do male because female mice menstruate, whatever, okay? All total BS. And when we start looking at differently, we're getting different responses. There are gerotherapeutics that are effective only in males or only in females. As far as the proteomics, so males have 600 proteins that are significantly increased, and females have less than half of that. Or we are calling it a stable proteome. So if we're going to develop biomarkers, either we have to do males and females differently, or we have to just include the ones that are common to both of them. But don't forget sex difference in everything you do, from the biology to treatment. Second thing, I didn't tell you the whole story, because those 1,000 people that we had, half of them were opus. Opus are offspring of parents with usual survival. They don't have longevity in their family. And you can see pretty much the picture that I showed you when I included everyone together before. And this is opal. Those are offspring of parents with exceptional longevity. And you just see with your eyes that this is much more red than this. Okay, in fact, those have 585 significant proteins, and those have 235 significant proteins. We know in our study that the opal are 10 years younger from every aspect than opus. And you see it here in the proteome. That still is not like treating somebody and see how it retreats, but you can see that those proteins are very significant in how they depict biological age. Okay, so those are, I think, the comment that I wanted to do on those biomarkers, and I'll be happy to touch upon more of it later. So, okay, so now I want to switch to gerotherapeutics, okay? And basically, you know, the best thing we could do is say, let's take a drug and over 5 years, see if it decreases mortality in population, okay? And understanding that in humans, longevity and healthcare and health span might be going together, not necessarily, we'll get there. Okay. Metformin, which you just had a really good review of, David, this old drug. By the way, sometimes I call it nutraceutical because it's an extract of the French lilac that was modulated somewhat, okay? Still needs a prescription. But the point with metformin that it extends health span and lifespan in animals. And the question, do we have any evidence of a picture like that with metformin? And the answer, we do. There is a study that included over 180,000 subjects where the data came from pharmacies and was adjusted for the patients, the doctors that treated them. It was around the UK. And basically, they looked at people who were diabetics and got either metformin or sulfonylurea and looked if there's different mortality. And you see there's half of the mortality with metformin. And by the way, the glucose control in the metformin was not as good as in sulfonylurea. So they had worse glucose control. It wasn't about the diabetes. But the question now was, how is it compared to people without diabetes? And to our great surprise, people on metformin with diabetes had significantly less mortality than people without diabetes. Let me say it even differently. The people that had metformin had diabetes. They were more obese than the control, and they had more diseases to start. And still they had 17% lower mortality. So our question is, let's take non-diabetic and give them metformin and see what happens to them. I mean, can we halve the mortality with the drug and kind of push it as a real good gerotherapeutic? And I think you said it right, and it's true that it's 100 years old, but there were other derivatives of the drug. Metformin is more than 70 years old. It's safe. And I think the point is it's generic and cheap. It's true, it's the cheapest drug in the market. If you get it from Mexico or Canada, it's even cheaper. You can get 400 pills for $20. And that's very important in what I'm going to tell you now. And by the way, this is the study itself. I made a cartoon out of it just to get you into it. This is the study itself. But I want to say just one thing. It's still not, it's a well-done study with lots of participants. It's not a clinical study. In other words, you know, how did the doctor decide to put somebody on metformin and or sulfonylurea? You know, what was the selection here? But it also shows you that the controls for either sulfonylurea or for metformin were overlaying each other. So, you know, you kind of have some internal controls in. Okay, but of course, what we care about is health span. And the nice thing about metformin, that there were clinical studies. Okay, clinical studies is placebo control or other treatment control. And also association studies that showed that metformin has really effect on everything. So the DPP showed that metformin delays diabetes by 30%. The UK PDS and other studies have showed that metformin decreased cardiovascular disease by 30%. Those are intervention studies. There are almost 300 studies of association of metformin with cancer, 30% less cancer over a 5-year period. Metformin in clinical study declines MCI, and in many studies, although all decrease Alzheimer. I just heard presentation last week from the UK, 200,000 patients comparing their Alzheimer's rate of people with new onset of diabetes and metformin and people without diabetes. And people with diabetes on metformin had 20% less Alzheimer's. In other words, there's a lot of literature of Alzheimer and metformin. And I showed you about lower mortality. So you must be saying, you've already shown that it decreased mortality. You're saying there are all those studies that are showing it does this and this on this disease and another disease. So what's the problem? And for us, it's taking metformin as a tool to solve a problem. Oh, before the problem, I forgot. I want to show you just something else. I'll get back to what we're trying to do, metformin as a tool. And I'm going to show you a rapidly moving slide very fast to make one point. Those are the hallmarks of aging down here in brown circles. Those are the hallmarks of aging. And the question, where does metformin, of all those hallmarks, where does metformin act? And metformin goes into the cell, it binds to the mitochondria, it inhibits complex 4, and on the left side here, it does a lot of things that I will call the metabolic. They even decrease insulin level and IGF level, and they target 5 of the hallmarks of aging. Okay, lots of literature, the paper, you can read the paper, I send you the paper. Okay, but there's other side because metformin inhibits also oxidation, and by that inhibits DNA damage, and has also actions that are not through mitochondria. And guess what? It targets all the hallmarks of aging. Now you must be looking at me and saying, we don't believe a word this guy is saying. And I want to make a point out of that because this is very common to all gerotherapeutics. We end up fighting about their mechanism, not realizing really that if you take an old cell or an old organ and an old body and make it younger, you're going to have lots of improvement. Okay, it doesn't mean that that's what metformin does initially, but at the end, all of those gerotherapeutics are affecting the hallmarks of aging. And also that is why those hallmarks of aging are interconnected. Okay, because you target one, you have improvements in others. Okay, so metformin is actually a potent anti-aging drug and directly or indirectly affect the hallmark of aging. So, okay, so back to why do we need to use metformin as a tool? Because really that metformin is not approved to target or to prevent aging. And if FDA doesn't recognize aging as a preventable condition, Healthcare providers don't have to pay, although with metformin it's cheap. But a lot of pharmaceuticals are on the sideline or on the fence. and they need a proof so they can jump in and develop better drugs, and we can do a combination of drugs and really have an impact on aging. And that's why I'm leading this study that's called TAME. It's not because I'm curious whether metformin is doing it. It'll do it. It's because I'm sitting with the FDA and discussing what should I do so that the FDA gives indication for aging. The study, basically, I'm just outlining, is taking people 65 to 80 that are not totally healthy, right? We don't want the people who will be centenarians. 3,000 of them, probably 3,500, double-blind placebo control. And our outcome is a cluster, okay? And so a cluster of those diseases that you see there. But let me explain, and let me put it very provocatively. I don't care what disease those people have, and I don't care what specific disease they're going to get. For me, it's all about aging. If your mother is diabetic and you're obese, you're going to get diabetic first, but it's really the time until you get any of those diseases. We want to move your health, okay, by preventing or delaying any disease that you are going to do next. That's what we're trying to do, which is a very new thought, okay? And it takes more than one time for people at the FDA to understand it. But we sat with the FDA. By the way, the FDA, we had diabetes there. The FDA doesn't like diabetes. We took it out. We recalculated our power. But also, as important is the NIA is paying us for a huge omic discovery on biomarkers, okay? Just like the methylation, just like the proteome, metabolome, microbiome, we're going to do it all because one of the important things for us is to have a real good study that will show you what does it mean to target aging from a biomarker perspective. I want to talk about some challenges that are also kind of important to practice. And the first thing that people ask me is, what age should we prescribe metformin? Because we keep saying that aging starts after fertilization, okay? The second is the question of, are all longevity therapies synergistic or additive? Is it possible that some of them are antagonistic? And I want to close with suggestions for other TAME-like studies. So this is a slide of a paper we recently published, and it's just one slide that intuitively depicts what we're trying to say. There's more data, and I'm just going to really ask you to look at the pattern. What you have here are people with high IGF across ages. What happens to them? And basically what it shows, that if you're young, it's good for you. It's good for mortality and dementia and diabetes and vascular disease and osteoporosis. High IGF is good when you're young, but when you're old, it's bad for you. It's bad for mortality, dementia, diabetes, vascular, osteoporosis. And by the way, if you're asking why not for cancer, because it's always bad for cancer. If you're young or old, high growth hormone is always bad, okay? But for other disease, that's how it looks like. And this is really supportive an hypothesis of aging that's called the antagonistic pleiotrope hypothesis of aging, that basically says things that are good for you when you're young can turn around against you when you're old, right? We need cholesterol to develop our brains and gonads and stuff, but if we have high cholesterol when we're old, we're screwed, okay? And pretty much the hypothesis with growth hormone is that we need to invest energy in growth in order to get to reproduction. But when we get to aging and we started this breakdown, we have to switch our energy, okay? We just have to lower the growth hormone and start dealing with the breakdown. This is the antagonistic pleiotraphy, okay? And I'm saying that because somebody last week, a 37-year-old guy, very athletic, really good body, says, should I start metformin? Okay. And I'm saying, well, it looks like IGF, lowering IGF is not good for you when you're young. The age of where we start gerotherapeutics is going to be important and has to have biological and clinical data to suggest it. Most of the trial on metformin have been done on people over the age of 50. There's some use of metformin in other cases, mostly when there's obesity, and obesity accelerates aging. But it's important to know that there's an antagonistic pleiotraphy, and it doesn't mean, senolytics is another example, okay? There's lots of senescent cells when you're old. And when we're young, there are not that many, but those that are there have actually clinical value, like helping with the bleeding control and stuff like that. So it's really important not to cluster aging as everything from birth when it comes to that. And I just talked about metformin. Okay, another example for synergism and antagonism is this trial. Charlotte Peterson had this great trial, the master trial, where she showed that basically she exercised elderly with and without metformin. Okay, and by the way, she got the grant because our hypotheses are that through AMP kinase, it'll be a synergistic effect. And in fact, it wasn't synergistic. Metformin blunted muscle hypertrophy in response to the progressive study in a placebo-controlled trial. And, before you say, I'm not taking metformin because it contradicts with exercise, it's actually important to read all parts of the study, including supplement 2, okay? Supplement 2 shows that with exercise, there's improvement in knee extension, maximum voluntary isometric contraction power, relatively strength. They were the same in both groups. So for me, it means, well, maybe the muscle was smaller, but every gram of muscle was stronger. Okay, so what I asked from Charlotte Peterson is to send me, she had biopsies of muscle, and I asked her to send those biopsies so I can look at the transcript. And what I found with the transcript is that exercise is in orange and metformin is in green, that indeed a lot of those transcripts decline with aging. Those are transcripts that are associated with mTOR. Okay, and mTOR is a nutrient sensing that we try to block with aging. Okay, it's the target of rapamycin. mTOR is the target of rapamycin. That's the TOR. So from an hypertrophic response, yeah, we see evidence that it decreased mTOR, which is what metformin is doing really from an aging perspective. On the other hand, metformin has improved some of those hallmarks of aging that are important, like the autophagy and the MAP kinase and cellular senescence and other things. So basically there are trade-offs, okay? So if you are young and bodybuilder and the muscle is important for you, then don't take metformin, okay? But if you're old, okay, and you want the muscle to be as effective, but you want it to be also protected from aging, then maybe you should do metformin and aging. And by the way, I'm on metformin. I'm taking metformin at night and exercising the next day in the afternoon. So I'm kind of building my own hypothesis, which is probably wrong, that you can do both of them. And lastly, how am I doing on time? Oh, great. Lastly, I want to tell you a paper that just came up this week. And actually, I'm really excited about this paper because we want to do not only one TAME study, but to do a lot so we get a lot of heat and move really quickly with gerotherapeutics. And we basically wanted to go from geroscience and see, look at FDA approved drugs and see if we have any potential for other drugs that are already in the market, FDA approved, to be a gerotherapeutic. And in order to get in this study, there are two things you had to do. You had to have at least one study that showed that animals live longer with this drug, and B, have an FDA approved. And there are not that many studies just to get into it. In other words, to show that there is longevity in animals. But there are all those ones. And we have a scale of 12. Okay, 6 points you get for preclinical data, right, that leads to drug development. And 6 points you get from human span and mortality data. So what do I mean by human health span? It's a drug that targets one thing and affects the other, okay? Not the same thing. If it's for diabetes and it's good for diabetes, that's not health span. If it's diabetes and it's good for kidney or heart, that's health span. The same thing for mortality. The mortality is not specific disease mortality. It's an overall mortality, okay? And you might be surprised, but it's the SGLT2 inhibitors, relatively new drugs, but in numerous studies, most of them in New England Journal of Medicine, they've really been incredible, and they really get the majority of points. The metformin gets only 11 points because there's you get 1 point for a study that was done in ITP. This is the NIH effort to show, and metformin was significantly better in male. It improved, rapamycin and metformin live longer than any other animals, but actually the trial itself was not significant. Other trials were significant. So it lost a point. Okay, it's really 11.5. But SGLT2 is really interesting. And I'll show you just one slide. This is from the ITP. It doesn't show so clearly here, but you see longevity, lifespans. Everybody is alive and they're starting to die. And you see the green line is extension of lifespan in those animals. And in humans, and I'm giving you just one study of the dapagliflozin that showed that all primary outcomes were much better. Renal outcomes were much better. Cardiovascular outcomes were much better. And any cause of death was much better. Okay, again, one drug that's given for diabetes. And by the way, many of those studies had non-diabetic people, okay? Because cardiologists have started using that, nephrologists have started using it, because all outcomes are better with this drug. So we have several options for intervention. I want just to say about TAME to make some other points. So I told you proof of concept, fine. I think we have that. FDA approval is crucial for us to move forward. And by the way, FDA didn't tell us, oh yeah, we believe that you're targeting aging, okay? When we're done, we can say we targeted aging. FDA can say whatever they want, but they agree that those outcomes of the major age-related disease are good outcomes. So that's fine. Even from investment perspective, I told you investors like the concept of hallmarks and they like the concept of TAME. That gives them, the horizons. Biomarkers, I told you it's important for biomarkers. It's a template for other drugs, like the one I just said. If any companies want to show how we target aging, that's how you do it. It's part of the wave that is happening now. The launch itself, which, delayed by COVID, will have an immediate impact on progress of everyone. It will be covered by the televisions and stuff. And I think what's important, it could be used because there's no healthcare providers that's going to give a big fight for a drug that is so cheap And will hope to show that the cost effect of treating is really high. But by the way, Andrew Scott took our data in his paper and showed that the value added by half of our patients, right, 1,500 on metformin, assuming that their health span will increase by 1 year, is $150 million. And the study costs $50 million. So the economy of someone should pay us to do this study.
Well, I'm going to circle back to the way you opened, Nir, and I think the most important thing you said is the most important, I think, message we need to deliver to the public, and that is that health span and lifespan are inextricably linked. I think that's pretty much what you said, and that if you enhance and extend lifespan, inevitably you're going to improve health span, because people get worried when you say that we're trying to allow people to age, enable people to age by 10, 20, 30 more years. They're worried that they're going to live with disease for that period of time. But this contraction of morbidity and an inextricable link between health span and lifespan is something that I think is critical, that most of the public doesn't understand. And quite frankly, I think a lot in the medical community don't understand that either.